So it's a great honor for us to welcome Charles Weissmann here. And I want to make this introduction in all seriousness in two parts. The first part will be rather general, and the second part will be rather specific. I think you've heard me say this before. In medicine, we have a term that certain doctors are referred to as doctors' doctors. And what is meant by that is when you really want a distinguished opinion on something, you don't go to a doctor, you go to a doctor's doctor. And Charles Weissmann is widely known in the scientific community, maybe not as a doctor's doctor, but certainly as a scientist, right? And it was for that reason that when we decided to build an institute in Florida, we needed a man like Professor Weissmann because this was a new venture. Nobody had heard of Scripps in Florida. Florida is not necessarily renowned for being a scientific mecca, and we wanted to make a statement about what we stood for. And what we stood for and continue to stand for in Florida as well as here is scientific excellence. And so in the entire world of science, we could think of no one better to be the emblem of what we thought we were going to be about than Charles Weissmann. Now in this institution, we did that once before, in a not identical, but a somewhat similar circumstance. And I tell you about this in order to reflect on how we think about Charles. Some of you may remember there was a time when the tobacco industry wanted to fund an institute in this town, and a medical institute, and of course, given the history of the tobacco companies, that was not necessarily the most enlightened decision, to say the least. It sort of was akin to when Dow wanted to build a napalm plant next to UCSD. Incidentally, you sit today on that Dow land that was donated to the institution because they could not escape the issues of the Vietnam War. So it's the Charles Weissmanns of the world and the Sidney Brenners of the world that we turned to when this institution wanted a symbol of incredible excellence. And we're very proud of both of those decisions, and both Charles and Sidney have remained close to us through the years. Now about Charles's scientific career. Charles' scientific career began in the Ochoa laboratory, where he began in what was the cutting edge area, of course, still is to some extent, in molecular biology, and that was the study of RNA phage replication. He spent 7 years with Ochoa, starting as a postdoc and winding up as an associate professor. Now, during that time, Charles invented the concept of quasi-species. A lot of people think they invented that. And that invention of quasi-species, which simply means that sort of the uncertainty principle of viruses. When you grow them up, because of the mutation frequency, you never get a uniform population. You actually get a species. And you can see how the thinking about quasi-species has influenced his career, later career in prion research, and I dare say will have influenced some of the things that we'll hear about today in terms of the fact that prions are not a single entity, but in fact are a species. So I asked Charles, or Charles said to me, he thinks that of all the things he did in his career, and of which there are many, many important things, this perhaps, in his view, was the most important. At any rate, in 1967, he left to go to Zurich to be the first director of the Institute of Molecular Biology. And there, he worked on the sequence of RNA, doubtless because he was working on RNA-phage. And in fact, his work on the sequence of RNA was instrumental to the thinking and ultimately the accomplishments of Sanger in the DNA sequencing format. Then I suppose most of you are here for one of two reasons, either to see how well he's aged, or you're interested in his, what most of us think is his most famous experiment. Maybe interferon was, but that was the 1985 cloning of the PRP gene in the scrapey system. And before Charles entered the scrapey world, not all would agree, but many would say that it was a rather murky black box. So you're looking and privileged today to hear from a man who has had an extremely distinguished career in science. We are proud that he's been associated with us for these many years. And you're about to listen today to a scientist. Charles?
Well, Richard, I think I have to start out by thanking you for having me come to Florida and participate in this great venture you launched, and allowing me to do science for 8 wonderful years. So thank you very much for that. And thank you for your kind introduction. I'm reminded of this, you know, the rabbi in Moscow who is being visited by the rabbi from Krakow, and he's introducing him to his congregation, and he says, this is Rabbi Cohen, well known for his piety, his knowledge of the Talmud, his knowledge of the Bible, his sense of justice. And then the rabbi tugs it, and don't forget the modesty. So actually, you forgot site-directed mutagenesis.
Okay.
So now, if I can turn this on, yes. So my talk is going to be on Darwinian evolution of prions, and I have to credit Jerry for inventing this very nice title, because I had a very complicated one about mutation and selection and so on, and Jerry cut it to the quick: Darwinian evolution of prions. And I would start out by telling you some elements about prions and what they do. So they cause what is called transmissible spongiform encephalopathies, or prion diseases. They're characterized by ataxia, dementia, and invariably death. And you find spongiform neurodegeneration and deposition of amyloid. There are long incubation times without symptoms, but once the disease breaks out, death ensues within a year or less. And interestingly, there is no immune reaction against the transmissible agent. The disease occurs both in humans, cattle. Currently, there is an epizootic in cervids in the United States, and also in mink. The human prion diseases, I'd just like to point out that there are so-called sporadic cases. In other words, like maybe one individual per million per year comes down with a disease called Creutzfeldt-Jakob disease, which is a prion disease, and which actually implies that prions seem to arise de novo, because these people don't contract it by contact with anything. Or there are also familial cases, so there are families which have a genetic, have a mutation which is dominant and causes a prion disease. But you can also acquire it. For example, there have been many cases where growth hormone was contaminated with prions. Growth hormone was up to the 90s. Growth hormone was made from human pituitaries. So if there, one of the pituitary glands was contaminated because the individual had suffered from a prion disease, the growth hormone would contain prions, and there were some 100-odd cases of young people coming down with prion disease. Well, I don't want to go into details except to point out that, interestingly, there is obviously this familial form of disease. What was discovered in the past 30 years is that the transmissible agent consists mainly, if not entirely, of a protein called PRP scrapey, and PRP scrapey is a conformational variant of a normal host protein, PRPC, a protein, glycoprotein, which is anchored to the cell surface by a GPI anchor. And here you see the structure, NMR structure done by Kurt Wüthrich in '96, which shows that there are the normal protein, the normal host protein, consists of 3 alpha helices, a very short beta sheet, and a large unstructured part here. Now, when a person contracts prion disease, this protein undergoes a conformational change, and this is a cartoon which are theoretical models which suggests that there is an aggregate of this protein, that it is rich in beta sheet structure. This has been established, that it is rich in beta sheet. Part of the molecule is susceptible to protease digestion, and a part of it is pretty resistant. So there are various models. None of them have really have been supported by evidence, because it is very difficult to study this PRP scrapey. First of all, it's difficult to purify, and it's aggregated. You cannot dissolve it. If you bring it into solution, it loses infectivity. So actually, the infectivity is bound somehow to the aggregated state of this protein. And obviously, difficult to do NMR, difficult to do X-ray crystallography. So this is still, the correct structure is still really not known. Now, how does this protein, so to speak, reproduce? In other words, why is this an infectious disease? And so the idea is that the normal protein can occur in various conformations, which may or may not be really the PRP scrapey conformation. It could be some related conformation. But the equilibrium is very strongly on the side of the PRPC, of the normal form, so that the steady-state level of this form is extremely low. However, when a certain number of subunits in the PRP scrapey conformation get together, and the number is assumed to be somewhere between 12 or 14 subunits, then you create a stable seed. So this is then stable, and then you can add on further subunits at a very rapid rate, and so this aggregate grows. It's eventually believed to be cleaved, and each of these can then continue to accrete further molecules of PRP scrapey. So altogether, because of this cleavage and the creation of new surfaces, you have a logarithmic increase in the amount of this infectious agent. And there's a lot of evidence which is in favor of this model, and it is widely accepted. It was first proposed by Carlton Gajdušek and Peter Lansbury many decades ago. Let me just quickly go over 3 lines of evidence which eventually led to the acceptance of this protein-only hypothesis, which was proposed by Stan Prusiner many decades ago. He ran into an enormous amount of opposition because nobody was willing to believe that an infectious agent could consist of only a protein. But eventually, 3 lines of evidence became available. The first is genetic, and I've mentioned that there are disease-linked mutations. So it turns out that the mutations occur in this molecule, in this PRP molecule. Then reverse genetic evidence, which is that if you knock out the PRP gene, then the animals, the mice, become totally resistant to the disease and do not propagate the agent. And third, biochemical evidence, and this is perhaps the most interesting one. There's a reaction called the PMCA reaction, protein misfolded cyclic amplification, and I'll tell you about that in a minute. So the genetic evidence was that a whole set of mutations in different parts of this PRP molecule were found, and each of these mutations shown here is a dominant mutation which causes familial disease. So this was, of course, a very strong piece of evidence which was brought forward by Stan Prusiner's group, Karen Xiao. The second line of evidence is the reverse genetic evidence I mentioned. We made this knockout mouse, this PRP knockout mouse. I should mention that when we undertook this work in the early 90s, we were sort of doubtful that, you know, it would be impossible to make such a mouse, because here was a protein which was mainly expressed in the brain. And as you know, Woody Allen said the brain is our second most important organ. So it seemed unlikely that a protein which is expressed mainly in the brain in all vertebrates, that you could delete that without anything happening. But as you see, this mouse is a homozygous deletion, and you can see by the intelligent expression on its face that it's a perfectly normal mouse. And interestingly then, when these mice were inoculated, the animals did not come down with a disease. They lived the normal life without any sign of replication of the prions, whereas the positive litter mates came down with the disease in about 170 days. Now, the third line of evidence is that it is possible to propagate infectious prions in a cell-free medium. So what Soto and his colleagues did was to take a brain homogenate as a source of PRPC, and then they added PRP scrapey to it. In other words, the conformationally altered, aggregated form of the PRP, which is associated with disease. And then they subjected that to alternating sonication and incubation, the idea being that sonication would cause these rods to break and create surfaces to which the PRPC could accrete. And so several cycles of this sonication and incubation eventually lead to the change in conformation of the PRPC into this aggregated form. This is a Western blot at the beginning of the reaction, and at the end of the reaction you see a tremendous amplification. And moreover, it is possible to carry out this cyclic amplification, then do a 1:10 dilution into a fresh brain homogenate, put it through the cycle, and you can do that for as long as you want, and you get a continuous production. You see that the level of PRP scrapey at the end of the incubation, even after every 1:10 dilution, the concentration remains the same, which means that you can virtually amplify this PRP scrapey in an unlimited fashion. And moreover, this product is infectious. So you can actually create infectious prions in a cell-free system. And a couple of years ago, 2 years ago, Wang et al. Showed that you could, instead of using brain homogenate, you could use 3 well-defined components, namely recombinant PRP made in E. coli, an anionic phospholipid, and poly-A. And if you went through this PMCA reaction with these 3 components, you could generate infectious prions and multiply them. Now, I should add that the poly-A seems to have a catalytic role rather than some sort of a structural role. But I won't go into the evidence for that. So the conclusion then is that these PMCA experiments definitively rule out the need for an informational nucleic acid, and also rule out a virus-like agent, because viruses, as you know, require a living cell in order to reproduce. And this reaction, this creation of infectious prions can be done with 3 well-defined components. So the next thing I want to mention is prion strains, because this is what has been interesting us for the past decade. So it turns out that many different strains of prions exist in mice, or also in hamsters, and also in humans. And this in itself was shown because these different isolates of prions show different incubation times, different deposition patterns of PRP scrapey in the brain, and a different lesion profile. But what is really interesting is that these different strains, many different strains, at least 20 different strains, can be propagated indefinitely in mice which have only a single genotype. So there's only one kind of PRP, only one PRP sequence, and yet this single type of PRP can give rise to many different strains. And I should also mention that strains may eventually switch or mutate when you transfer them from one host to another, say from mouse to hamster and back to mouse, or they can remain the same. Now, this shows you a picture from a paper by Prions group. It shows 3 different mouse strains, and what you see here is the deposition pattern of PRP scrapey, and you see that characteristically these depositions differ for the different strains. It is believed, and I think this is a correct belief, that the strain properties, or in other words, the strainness, is enciphered by the conformation of the PRP. So in other words, you don't only have the normal conformation of the pathological one, but you have dozens of different pathological conformations, each of which confers the properties, strain-specific properties. And there is evidence, which I won't go into now, which are based on the protease resistance, infrared spectrum, hydrogen deuterium exchange patterns, which show that different PRP scrapey from different strains have different physical properties. So classically, it was a very difficult and long-term project to distinguish strains, because you have to infect mice and observe what the incubation time is and the pathology and so on, and it took 1 to 2 years to differentiate prion strains. And eventually, when I left Zurich and then when I was in London for a couple of years, we devised an assay which is based on a cell assay. So instead of injecting animals and measuring how long it takes for them to get sick in order to quantitate the prions, we devised a cell-based assay, which was based on finding a cell line which is very sensitive to prions, and on the observation that it was possible to identify single infected cells by virtue of the fact that they accumulate PRP scrapey. And so the assay was simply this. We would take an infected solution, an infected brain homogenate, make a serial dilution, then infect cells with these different dilutions, incubate them for 3 splits to allow the cells to become infected, and then we would plate out 20,000 cells on a 96-well plate, on the membrane of a 96-well plate, identify infected cells, and there was a dynamic relationship between the concentration of this infected solution and the number of infected cells. And this is what the infected cells look like. The counting is, of course, automated. And what you find then is a dynamic response. What we plot here is the number of infected cells against the dilution, or I should say in this strain, against the concentration. And you see this response, so the more concentrated the solution, the greater the number of infected cells which you can count. And then we can define and measure for the level of infectivity, which we call the response index. and which is simply 1 over the dilution, which gives rise to an arbitrary number of infected cells. Let us say we often use 300 cells, and so we then can express the level of infectivity by this parameter, by the dilution at which you attain 300 infected spots. So this worked very nicely for 2 strains, for RML and 22L, but when we looked at different strains, the cells did not respond. So my colleague Suki Mahal then started screening many cell lines to find whether you could get cell lines which would be responsive to other strains. And at the end of the day, she isolated 4 cell lines, PK1, R33, which are derived from a neuroblastoma cell line, LD9, which is derived from fibroblasts, and CAD5 from a neuronal tumor. And she found that we could readily distinguish 4 different strains, which are listed here, RML, ME7, 22L, 301C. We have since expanded this, but this was the first time we were using this so-called cell panel assay to differentiate between different strains, and that only took 2 weeks rather than 2 years. And so what Suki then found is that the different strains show different patterns on the different cell lines. For example, the strain RML can readily infect CAD cells, PK1 cells, and LD9 cells, but cannot infect R33 cells. The strain 22L infects all 4 strains, albeit the R33 less effectively, but all of them can be infected. ME7 will only infect LD9 and CAD5, whereas 301C will only infect CAD5. So you see that you can set up a sort of a matrix, and if you find that your sample can infect all 4 cell lines, then it is compatible with being 22L. If it infects 3 cell lines, RML, 2 is ME7, and 301C if only CAD cells are infected. Of course, you may have other strains which show a similar pattern, but right now we're dealing with 4 strains, and these 4 strains can be differentiated. So just a quick comment, we found that 2 compounds, swainsonine and cefunensin, which are inhibitors, turn out to be inhibitors which are specific for certain prion strain cell combinations. So these inhibitors act at different positions, at different steps during the biosynthesis of the glycans. And I just mentioned this, that these are inhibitors of glycosylation. I can't go into the reason right now why they act that way, but the fact is that these inhibitors cause misglycosylation of prions in certain cell lines, and these misglycosylated prions are degraded much more rapidly than they would be normally. And so this happens in some cell lines for some strains, and it's an additional diagnostic implementation. And just to show you here for RML on this PK1 cell line gives you 3 logs of inhibition. You see there are 3 logs of inhibition here. Whereas 22L on the same cell line shows no inhibition at all. This is just because we're going to use this swainsonine as a tool in what I'm going to tell you. So the first question we decided to tackle is whether a prion strain, upon transfer from infected brain to cultured cells, whether it remains the same or whether it undergoes some sort of change. And so in this context, we are now going to talk about mutation, meaning an heritable change of a prion strain. But in this case, it implies a change at the protein level, most likely a conformational change. And so when you transfer 22L prions from brain to PK1 cells, they change their cell tropism. So this is 22L after propagation in brain, and as I mentioned before, all 4 cell lines can be infected, R33 a little less well. Now, if you propagate 22L prions in these cells for many passages and then test their cell tropism, you find that they have become completely sensitive to swainsonine. So you see here, swainsonine has virtually no effect. This PK1 is the light blue. PK1 plus swainsonine is the dark blue, so virtually completely resistant to swainsonine. But after being propagated in PK1 cells, they become totally susceptible to swainsonine. And they are no longer able to infect R33 cells. So you might argue, well, that's because the prions are made in a different environment and they're made of different components and so on. So we thought, well, let us see what the kinetics are of this change. If it is a sudden change, in other words, as soon as you put these prions on cells, they change properties, then it may be likely it's some other component which is causing this problem. But maybe it takes time for this to happen. And so so this is the question I'm posing: is the change immediate, suggesting that it is just simply because other components are involved, or is it gradual? And the answer is shown here. So this is brain 22L. We're just looking at 3 cell lines now. The PK1 cells in blue, the PK1 plus swainsonine in red. You see completely resistant in brain and competent to grow on R33. Now, as you start passaging them in PK1 cells, as soon as you go into cells, you already see a difference. They're starting to become sensitive to swainsonine, and with every succeeding passage, they become more and more sensitive to swainsonine until they're fully sensitive and incapable of growing on R33 cells. So the idea then was, because our awareness of this quasi-species nature of viruses, the hypothesis was, well, maybe prions are also a sort of a mixed population. You have a major component now, which is dark blue in the brain, but you have a scattering of variants, yellow, green, red, the red one being perhaps one which is resistant to swainsonine. So, the red one being one which is capable of reproducing in PK1 cells. And so we think that as you switch over now to PK1 cells, this yellow background, you start having an advantage for the yellow variants. And as you continue to passage them, the yellow becomes dominant and all the others become minor components. So you end up again with a quasi-population, but the dominant species is now one which originally was a minor species. Now, when you do the opposite, you take these prions from the infected cells and put them back into brain, the exact opposite happens. They now reacquire the original properties of 22L. In other words, they grow on R33, they become swainsonine resistant, and they resemble the original 22L. So this is a process which is reversible, as you might expect if you end up with a sort of a quasi-population. So in short then, the cell panel-acid characteristics of the 22L population gradually change when transferred from brain to PK1 cells, but when returned to brain, the population gradually reacquires the original properties. And the hypothesis, as I said before, is that a cell-tropic variant is selected in cells and a brain-tropic variant is selected in brain. Then we also found that prions can acquire drug resistance. The experiment was a fortuitous observation. We were growing in 22L infected PK1 cells, and we wanted to see whether, if we added swainsonine, whether we could then cure the cells of prion infection. So we expected that they would be cured, but as a matter of fact, this did not happen, but they became resistant to swainsonine. So the experiment was either propagating these these 22L infected PK1 cells in the absence of swainsonine, or we propagated them in the presence in the presence of Swainsonine, or we incubated them first, grew them first in the presence of Swainsonine and then in the absence. And when we did the experiment, what we're looking at now is the response to PK1 and here inhibition by Swainsonine. So if you simply grow them without adding Swainsonine, they just remain what they are, sensitive to Swainsonine. But if you grow them in the presence of Swainsonine, what happens is immediately after Swainsonine, you get a dramatic drop in the infectious titer, but then pretty soon it picks up, and everything you see now is resistant to Swainsonine. But if you, at this stage, go back to cells in the absence of Swainsonine, then eventually, after several splits, you get a population which is again Swainsonine sensitive. So what this means is that under normal circumstances, the Swainsonine sensitive prions grow faster than the resistant ones, but of course in the presence of Swainsonine, only the resistant ones can survive. So, what we think is happening is now here in a scheme. So when you grow in the absence of Swainsonine, nothing happens. Here you have the dominant prion is the yellow one, it stays yellow. When you grow it in the presence of Swainsonine, almost everything dies off, but there are some Swainsonine-resistant survivors, and they then take over the population. And when you go back to the absence of Swainsonine, these yellow ones end up dominating the picture again. So then 22L, as you might remember, originally the 22L in brain starts out by being resistant to Swainsonine, and it only changes when it goes into PK1 cells. So the question was, does a strain which normally is sensitive to Swainsonine, the brain sensitive to Swainsonine, can you nevertheless force it to become Swainsonine resistant? And so we start out with RML. So RML, when you look at the pattern of RML, you see that it is totally, that it has this inhibition, in this case, I think about 1.5 logs inhibition by Swainsonine. If you now grow them on PK1 cells, you infect PK1 cells and expose them to Swainsonine, everything disappears. So they are, as you might have originally expected, they cannot grow in the presence of Swainsonine, but you also don't get any resistant mutants, and of course when you do the assay, zero, there is absolutely no response. But then my colleague Anja Oelschlegel started looking at other cell lines, and she found one among several, which we call the PK10 cells. When you introduce the RML prions into PK10 cells and expose them to Swainsonine, you now get Swainsonine resistance. So, as you see here, they're completely sensitive, resistant to Swainsonine. And if you put them back into the mouse, you remember in the previous experiments, when you went back to mouse, you ended up with what you had started with. So there was this reversion back to the original properties. But in this case, interestingly, when we harvested the prions from the mouse and tested them, they had changed their properties in a very dramatic fashion, in that you now, if you test them with this other inhibitor, kifunensin, you see that the original RML is completely sensitive to kifunensin. But once it's gone through this procedure and you end up with this mouse, it's completely resistant to kifunensin. So in this particular case, we somehow have driven these prions into SWA resistance, and that also entails resistance to this other inhibitor of glycosylation. And this is now apparently a stable new strain which came out of this experiment. So it's a different situation from what we had before, and I'll come back to what this might mean in a minute. So after having found that 22L populations in PK1 cells can become Swainson resistant. The question is, are these Swainson resistant variants already present to begin with in this population, or do they arise during the treatment with the inhibitor? And so the experiment we did was to take these PK1-derived 22L prions and screen them for pre-existing resistant variants. The way this is done is to infect cells with this 22L supernatant. In other words, the supernatant of these cells contains prions. We infect cells with the supernatant, and we divide the cells into two portions. One is infected and grown in the presence of Swainson, and the other in the absence of Swainson. And what we do then is because you don't infect all cells, you only infect a fraction of the total cells, what you want to compare is the number of Swainson-resistant variants compared to the total number of prions which you started out with. And the way you do that is, I won't go into the details, you grow them over 3 splits, and you add different numbers of cells to each well, because you expect few Swainson-resistant variants. So you use here 1,000 cells per well, here only 8 cells per well. You count how many Swainson-resistant prions you had and how many total. And if you do the Poisson distribution calculations, you find out that about 0.5% of the prions you start out with were Swainson-resistant to begin with. So the conclusion then is that these lysids contain about 0.5% Swainson-resistant prions, that they were present prior to the selection. And then the important point is this. 22L strain, which we use in our laboratory, had been biologically cloned by the people who isolated the strain. Biological cloning means that you infect mice with a high dilution of prions, such that on average there's only 1 prion per 10 mice. And when you do that for a couple of cycles, you come up with what is presumed to be a cloned strain, 22L strain. So the original strain, 22L strain, must have acquired. It must have acquired a resistant prion somewhere between the time it had been cloned and by the time we did our experiments. And therefore, we decided to determine what the kinetics of appearance are of Swainson-resistant prions. And the way we did that was to prepare infected cells at a very high dilution of prions, so that we now clone these prions in the PK1 cells. And so only 1 among 10 or 20 cells will contain a prion. And when we now we now propagate this clone, this infected cell, and we assay the prions, and we find that they're totally Swainson sensitive. So the idea is that the bulk of the prions that we're looking at were sensitive, and therefore this single prion we had here was also sensitive. Then we grew them for another 22 doublings, either in the absence of Swainson, and they continue to be sensitive, or in the presence of Swainson. And then it turns out that obviously somewhere along this line here, there must have been a mutation generating a Swainson-resistant mutant, which then, upon challenging with Swainson, gave rise to a resistant population. This experiment was carried out with many clones, and the conclusion of the experiment was that 2 out of 8 clones acquired mutants, SWA-resistant mutants, within 31 doublings. One of the cloned populations acquired, one of the populations acquired a resistant mutant within 53 doublings, and making many simplistic assumptions, we calculate that the approximate mutation rate is about 10 to the minus 6 per doubling. So very, very rough number. So this is what we are proposing then. We take this population, we use individual prions, which are all of the blue kind, to infect the cells. And as we propagate the cells, a resistant, or in this case a red prion will arise. Here perhaps nothing happens. In another couple of transmissions, you now acquire a yellow mutant here and a red mutant, and perhaps here a green mutant. So at the end of the day, you have these quasi-populations. And I should add that in such a quasi-population, you have a constant selection against the variants, a constant rate of generation of new variants. So you have a dynamic, steady state of these different entities. And so what do we think how do we think does a mutation come about? Well, we think that somehow the PRP scrapey is an aggregate consisting of a conformationally altered PRP, as you see here. I already mentioned that it's presumed that these aggregates can be cleaved to generate smaller aggregates. And now the question is how, you see, if you have a large aggregate in a conformation which is, let us say, energetically unfavorable, it is frozen into this conformation by virtue of its being part of a large aggregate, just as molecules in a crystal are frozen in a particular conformation. And what we hypothesize is that if you have smaller aggregates, there is a possibility of a small conformational variation. And so if you have this thermal fluctuation, you may change a little bit this conformation. And when this happens, and you add further PRP molecules to it, which assume this conformation, then you fix this altered conformation. So this is just a way about thinking what mutation might mean in the case of prions. But this is, of course, as you realize, totally hypothetical. Now, I've shown you that prions can adapt to different environments, cells or brains, different cell lines. They can adapt to the presence of an inhibitor. And here's an experiment which we carried out in collaboration with Michael Oldstone, which shows that prions can also adapt to a modified substrate. And the modified substrate is this. Cheeseborough and Oldstone, their colleagues, generated a PRP gene which was truncated at position 231. Now, you remember PRP is GPI linked, and that means that during maturation, there is a cleavage at position 231 with the addition of a GPI anchor. And the deletion Cheeseborough et al. Made was to delete this part which is required for the addition of GPI anchor. As a result, instead of having the PRP anchored to the membrane and then exposed eventually at the surface of the cell, once you remove the GPI anchor, the PRP is released into the medium. Interestingly, when these mice were infected with prions, with various strains of prions, somewhat surprisingly, it turned out that they were infectable, that they made infectious PRP scrapey, but that the distribution of the PRP scrapey in the brain was very different. So in the wild-type mouse, you see this diffuse brown presence of PRP scrapey sort of within the cells all over this region. But in the case of the GPI minus mouse, the one which no longer can anchor the PRP scrapey was found extracellular in the form of aggregates between the cells and, as it turns out, around the capillaries of the brain. So this suggests that the transition of PRPC to PRP scrapey in the case of the GPI minus mouse no longer occurred intracellularly, but extracellularly. And so the question arose, what does that do to the fidelity of strain replication? And it turns out that if you look at the normal RML grown in wild-type brain, you have this pattern that, as I said before, all these three cell lines, these two cell lines can be infected. PK1 shows about 1.5 logs inhibition by Swainsonine, and the RML prions are incapable of infecting R33. However, the prions you get out of the GPI minus mice, they are resistant to Swainsonine. They can infect R33, and they've obviously changed in a dramatic fashion. Is that because the PRP scrapey itself no longer contains this anchor, or is it for some other reason? And so the answer can be obtained by taking these prions and putting them back into wild-type mice and seeing whether you get back the original RML or something else. And Suki Mahal did this work. It's now in press, not yet out. So what she did was to take the prions that had been passaged through the GPI minus mice and returned to wild-type mice, and she found that these prions have now become resistant to two inhibitors to which they were susceptible before. And you see this here. The RML itself is highly susceptible to an inhibitor castanospermin and very susceptible to kifunensin. Once you go through the GPI minus and into wild type, the prions are now resistant to kifunensin and much less susceptible to castanospermin. And after passaging twice and even 3 times through wild type mice, this pattern doesn't change. So in this case then, again, a new stable strain has emerged, which we call the Scripps, Florida laboratory strain. So this raises an interesting question. If you start out with a strain which is stable in mouse brain, and you go through this transition in GPI mice, and you come out with something different, and this is also stable in mouse, why is it that it doesn't change back into RML, or conversely, why does RML not spontaneously change into this mutant SFL? And we propose the following explanation. So what you see here is an energy diagram, and you see that there is a major activation energy barrier between the RML, the postulated RML structure, and the SFL. So in the case of RML, you have all these small wells which are populated because it is a quasi-species. So you have all these different, slightly different conformations which can easily interconvert. And but you cannot convert it to SFL because there's a very high activation energy in wild-type mice. And so what we postulate is that for reasons which we don't understand, the energy diagram in GPI minus mice is different. It has a lower activation energy between the RML and SFL and a higher stability in the SFL. So that within the GPI minus mice, you can understand that it's now populating these energy wells. But when you put them back in wild type, they are now trapped in this compartment because, again, you have this high energy barrier, but now they're trapped in a different compartment. So this is, shall we say, a reasonable explanation to explain what is really not so easy to understand, namely this stability of two strains in the same host. So in summary then, prion variants arise in cloned populations, presumably by what we call conformational mutation. The number of possible variants is immense. And because you have a huge number of possible conformations, and each conformation can be frozen in a aggregate. So right now we know of at least 30 mutations of mouse prions, and many variants may still be found. Distinct prion populations are selected in different environments, and prions, although consisting of protein, exhibit Darwinian evolution. And I should stress that this work was done largely by Suki Mahal, who generated the cell panel assay, observed the strain switch, did the work on the GPI- mice, which I've just mentioned, and Jai Li, who did the Swainson-resistant prions and the very elaborate cloning experiment I told you about. Sean Browning discovered this Swainson inhibition, and it's very interesting what the mechanisms were. Can't go into that now. And Anja Oelschlegel, she studied the prion population heterogeneity and the SWA-resistant RML experiment I told you about. And these people were helpful, and this is the end. Oh, wait a minute. The end of the work, the end of my group, and the end of my tenure in Scripps, which has been a marvelous experience. Thank you.